This is Robert Sapolsky, and you're listening to Father-Offspring Interviews.
Hi, welcome back to Father-Offspring Interviews, episode 56. Before we get into content, we just want to let you all know that this video component is now available on Spotify as well, so more platform flexibility for your viewing. We're going to start today with two related questions. First, Giada from Italy says, what is your opinion on Ozempic and similar drugs like Mounjaro, which are based on semaglutide? I've heard some discussion about the potential use of these medications to treat addictions, such as drug, alcohol, or gambling addiction. And Natasha from Oregon asks, what do you make of recent pilot studies on GLP-1 drugs for obesity, binge eating, and now substance use disorders?
At last. Ozempic and its siblings, its related molecules. As of about 3 years ago, Ozempic and related semaglutides were without question the most exciting molecules in the universe. And then, inconceivably, now a few years later, they've become even more exciting because of findings alluded to in those questions. Okay, Ozempic, semaglutides, these are drugs that now famously suppress appetite. They may be the most exciting means of reversing obesity on this planet. Everyone has heard about it. A small privileged subset of humans can afford it. What's it about? What's it about in terms of suppressing appetite, weight loss, and what's this unexpected realm of additional actions that it may have? Okay, Intro 101 on appetite. Appetite all circles around part of your brain, the hypothalamus, and a sub-area of it called the arcuate nucleus. This is where your brain, your hypothalamus, is deciding, are you hungry or are you satiated? And the way it is doing that in our simplest first pass is there's messengers coming from your gut, from your pancreas, from your fat cells, telling you about your nutritional state. And there's one hormone in particular called ghrelin, which comes when you are hungry. When you are hungry, what triggers its release from the gut? When your gut is empty, when it hasn't been digesting anything for a while, ghrelin is secreted and gets up to your hypothalamus, your arcuate nucleus, where it then stimulates eating. Flip side, when you've eaten a lot, when your fat cells are full, or when your gut is full and distended, you secrete completely different hormones that have the opposite effects that cause signals of satiation. First of those hormones that come from fat cells were the most amazing gloried molecules on Earth about 20 years ago, because the thought was they were going to completely solve obesity. And this is a hormone called leptin. And leptin was so exciting and so popular until along came these classes of hormones released from the gut, glucagon-like peptides. GLP, the major one, GLP-1, glucagon. Glucagon is this obscure hormone that comes out of the pancreas, and it's been around forever. And suddenly GLP-1, the glucagon-like peptides, are like more famous. It's like an Elvis impersonator selling more albums than Elvis himself. I have a very soft spot for glucagon personally, so it being overshadowed by GLP-1. What does GLP-1 do? Much like leptin, it is a signal of satiation. In its case, it is released from the gut when it is distended with lots of food, and it also gets into the arcuate nucleus and inhibits feeding there, increases the sense of satiation. Okay, that's wonderful. But now comes along the imitator of the Elvis imitator, which are the semaglutide drugs, the drugs that mimic GLP-1 drugs, that bind to GLP-1 receptors in the brain and activate them as if they were GLP-1 itself. And what you have is seemingly a very simple circuit. You haven't eaten, you're hungry, ghrelin makes you eat. You've eaten a lot, you secrete leptin and GLP-1, and it tells your hypothalamus to no longer be hungry. And what are semaglutides like Ozempic doing, they are looping their way in there when you're not secreting enough of your own semaglutides to generate a satiation signal. And they come in there and they mimic it and fake out the receptors, and you have a sense of fullness, a sense of satiation. And this is the basis of this tidal wave of people using Ozempic and related drugs, and it's having remarkable effects. It's causing, for the first time in decades, in the last few years, a decrease in the rate of obesity in the United States. And interestingly, informatively, it is mostly being carried by loss of weight among people who are middle class or higher. In other words, if you're lucky enough to have enough money to buy the stuff, or you're lucky enough to have insurance to sort of cover the unconscionably high costs of the drug, you're great. Okay, so it is terrific for people losing weight, and along with it now comes all sorts of downstream advantages of that: decreased likelihood of type 2 diabetes, decreased likelihood of various cardiovascular diseases, strokes, the very preliminary data suggesting, as one might expect given those really great effects, extended lifespan. Okay, so that is our very simplistic first world of making sense of this, which is you get signals from your body saying no food, hormone comes in, makes you eat. Got signals from your body saying you've eaten a lot, and among the hormones released, GLP-1 comes and signals in your hypothalamus that you are satisfied, you are satiated. And what do drugs like Ozempic do? They fake out the receptors for GLP-1 in the arcuate nucleus in the hypothalamus and make it believe you are satiated, and thus you eat less and the world is a much more benign place. Let's make this more complicated now in two ways. The first one, let's consider this mostly a sideshow. It turns out that GLP-1 is also made in the brain, and people are just beginning to sort that out. But for the moment, all we care about is the world of distended guts releasing GLP-1 and making your hypothalamus feel satisfied and satiated, or GLP-1 type mimickers like Ozempic accomplishing the same thing. But then it turns out there's GLP-1 receptors in lots of other places in the brain. And when you look at it, it begins to make sense from the standpoint of what goes into regulating appetite, regulating satiation, and you see all sorts of interesting outposts. For example, part of the brain called the insula, the insular cortex, which we've talked about many episodes ago. Insular cortex, it's incredibly interesting in humans. In this case, we're instead falling back on the boring thing it does for all mammals out there, which is the insular cortex activates when you eat some disgusting rotten food. It triggers you to spit it out. It makes your stomach lurch. It protects you from toxic food. It mediates gustatory disgust. And what happens is that GLP-1, when your gut is distended and you are satiated, GLP-1 is also having effects in the insula, making food seem maybe even disgusting at that point. My God, I couldn't eat another bite. So that has something to do with it. GLP-1 works in your olfactory system and makes food smell less appealing, less palatable. GLP-1 works in your hippocampus having something to do with memory, and that's mighty relevant. Feeding, no longer feeding, is very intertwined with memory. If you are, some foraging bird, you've got to remember where you've hidden food in various places in the forest. Hippocampus is very relevant, activated in that regard when you were hungry. When you were trying to suppress eating, I don't know, that same bird will think it's only 3 months till bathing suit season or something, so don't eat that extra worm. And in this case, it's got to remember the time of year. So memory comes into it also. Lots of different brain regions. And thus, what we're seeing here is appetite, satiation winds up being far more than just turning an on or off switch in your hypothalamus. on or off in your hypothalamus is just like a simple thermostat setting. As soon as you're bringing in memory and olfaction, and if you are deciding the food smells disgusting, or if you're deciding you were disgusted with yourself from overeating, you are now bringing in all sorts of other outposts in the brain, and GLP-1, and thus Ozempic, is having effects in these areas. All right, but now we get to the third level, the most interesting one that brings all of this up to the questions that were raised, which is GLP-1 receptors are also found in a part of the brain related to the neurotransmitter dopamine, reward, anticipation. For neuroanatomy fans, the ventral tegmental area, which sends projections to the nucleus accumbens, which sends it on to your limbic system and frontal cortex. And these turn out to be target sites for GLP-1 action and Ozempic action as well. And what it's doing is suddenly explaining one of the first interesting findings about Ozempic. Not only does it make you feel full, I am satiated, it has a negative feedback signal at the hypothalamus, it's also tapping into reward and anticipation pathways. And as a result, food seems less appealing. You're less motivated to eat it. You have less cravings. It's not just affecting your gut talking to your boring old hypothalamus and saying you're full. It's also affecting anticipation of food, the pleasure you are likely to get, the pleasure you are no longer feeling because you've habituated to it. We're getting a much wider scope here. And here we get into the realm of these questions, which is in the last few years, all sorts of reports coming out that not only does Ozempic and its related friends, not only do these semaglutides decrease appetite for food, they decrease cravings for all sorts of addictive behaviors. And this has taken the world by storm. What's the evidence at this point? Exactly as Natasha brought up, these are pilot studies at this stage. They are just hints at it. There have been, to my knowledge, no well-controlled, double-blind studies designed beforehand with humans. This is what we know at this point. You take animals, lab animals, rats, mice, monkeys, and what you show is when you give them Ozempic-like drugs, their interest in drinking alcohol goes down. Rats and mice and monkeys and humans are happy to drink alcohol under all sorts of circumstances and become addicted to it, become having a need for using it to excess. And what you see is drugs like Ozempic decrease alcohol craving. In mice and rats. Okay, how about humans? And here we get into a very squishy literature, which is one where scientists have done some very quantitative, hard-ass analyses of some very squishy, unquantitative data. They've done these massive AI trawls through social media and looking for conversations people have about Ozempic and looking for talking about alcohol, talking about cigarettes, talking about caffeine, talking about all sorts of things that we get addictive about, and looking what are people reporting in these. And these are tens of thousands, hundreds of thousands that are scanned through by this AI and can come back and give numbers, which is roughly 25% of the people out there talking about Ozempic and smoking cessation, alcohol, caffeine, compulsive shopping, compulsive gambling, that about 25% of them are saying, this is miraculous, all my urges for it have disappeared. So that's impressive. But still, no one has sat down any individuals who are starting to take Ozempic for obesity and showing that it's affecting these cravings. But we tap into one other domain where there's insights, which is Ozempic and the related semaglutides have actually been around for a while as diabetes drugs. And there's a literature by now showing people who were taking Ozempic for their diabetes problems, what you see is decreased incidence of alcohol abuse, decreased substance abuse in general, all of these themes coming through. So it's beginning to look like that's the case. So, what do we make of that? Whoa, this is your drug that's talking to your hypothalamus, and it's faking your hypothalamus to thinking, my gut is so distended, I couldn't possibly eat another thing. And you gamble less. You shop less on Amazon, online. You have all sorts of substances of abuse that you abuse less than you were doing. Where's this coming from? And I think what we're seeing is this theme with the brain. Our fancy primate brains are really lousy at dealing with metaphors. Our brain has a whole lot of trouble telling the difference between the literal and the metaphorical. Back to the insular cortex, something we've talked about before. Yes, in a rat, it tells you this is disgusting food. In a human, it tells you this is disgusting food you've just tasted. In a human, it tells you you're looking at a picture of somebody eating a disgusting food. Oh, we're an abstract species, we can do that. In humans, it also tells you if you were contemplating somebody having carried out an act which is morally repulsive and morally disgusting. Whoa, the insula activates if you've just eaten some rotten food or you've just heard about some horrendous, awful thing that has been done to somebody. Neurons in your insular cortex cannot tell the difference between literal gustatory disgust and moral disgust. Another part of the brain, your anterior cingulate, and we've covered this also. What it's good for is it helps you interpret when you're getting pain signals in your body. Is this scary? Is this good news, bad news, whatever? But then go look at your loved one in pain, and your anterior cingulate activates as well. Whoa, neurons in there can't tell the difference between the literalness of someone who's just chopped your toe off and the metaphor of you feeling somebody else's pain. So there's this theme over and over that the brain really confuses the metaphorical with the literal because, you just got these old brain parts to use and you just kind of wing it when new stuff like moral disgust gets invented. And I think what we're seeing here is as soon as we see drugs that mimic GLP-1s, as soon as we see them not just in the hypothalamus and not just in the olfactory bulb, but in your reward areas and in your frontal cortex, what we're tapping into is this metaphorical sense of we humans can have a hunger for all sorts of things. We can hunger for food, we can hunger for drugs, we can hunger for sex, we can hunger for money, for all sorts of things. You could hunger for having the greatest, rarest stamp collection on earth, if that's your particular dopamine makeup. We have a whole lot of very abstract hungers. And I think what this is telling us is this is another realm where your brain, your dopamine system, is falling for, you're not actually having a real hunger that your brain would understand at the level of GLP-1 when you were just hungering for like a great novel to read over the weekend. Actually, maybe that is the case. And I think what we're seeing as soon as GLP-1 and drugs like Ozempic are getting up to reward areas, we're tapping into the fact that we hunger for a lot of things. We have cravings for a lot of things. And in a funny way, your brain can't tell the difference between hungering for food and hungering for gambling away your life savings in Las Vegas. Hunger is a very generalized, abstracted thing in the brain. And unexpectedly, this drug that very specifically mimics this hormone is tapping into metaphorical hunger and metaphorical satiation.
To wrap up today, Dan from Ohio says, is there any biological evidence on the effects of the degree of inbreeding versus outbreeding on human psychology and culture?
Inbreeding, we all know about the adverse effects of inbreeding. You get the Habsburgs, you get the pharaohs marrying their sisters. You've got all these examples where, you're going to have babies with tails and 6 fingers. The general term for it is that you see inbreeding depression, a decrease in the fitness of individuals, humans or otherwise, that are the outcome of inbreeding. And in the human literature, for example, you could see degree of inbreeding, elevated levels of inbreeding, and you will drive down birth weight at birth. You will increase likelihood of cardiovascular diseases, abnormal BMIs, abnormal life expectancies. Yeah, inbreeding depression. It's bad for you. It's absolutely clear. What's the mechanism for it? And this is, some ninth grade biology stuff. You can have a genetic trait that's in a mild form, heterozygotic, or in a full-blown version, homozygotic. And there's a lot of diseases where when you carry the mutation for it in only the partial heterozygotic form, nothing goes wrong. It's perfectly fine. And in fact, there may be some advantages to it. And it's only with inbreeding that the odds increase that you are now mating with someone who is also heterozygotic for that. And an outcome of that is a homozygotic offspring who gets the full-blown version of the disease. So a very clear message coming out of that: do not mate with your close relatives. Nonetheless, some degree of inbreeding has been found throughout history in various cultures, and addressing the question, are there any predictors? Cross-cultural studies, one from 180 cultures from around the world, looking at the average degree of inbreeding. And what you see are two logical predictors. You see lots of inbreeding in populations that historically have been isolated, so your options are limited, and or populations with a high historical load of infectious diseases, pathogens. You don't welcome outsiders into your community because who knows what diseases they're bringing with them. And in effect, you then have a very narrow pool of potential mates. So we get a very clear punchline out of that: avoid inbreeding and inbreeding depression, and it's not a good thing. Don't do that. But there's a very interesting thing going on that pushes in the opposite direction. Back a bunch of episodes, we talked about sociobiology, the evolution of social behavior, and we saw that there were three basic pillars of evolution of social behavior: individual selection. Animals behave to maximize the number of genes they leave in the next generation. Kin selection. Sometimes you can do that by helping close relatives reproduce as much as possible as a function of how many genes they share with you. And the third leg, sometimes it makes sense to cooperate with non-relatives if they cooperate back. Okay, so this kin selection part, there's all sorts of advantages to cooperating with kin. The closer, the more reasons there are to be cooperative. So now we look at one aspect of the battle of the sexes and lots of species: intersexual conflict. What does that look like? You look at a species that does not have monogamy, say hamsters. Hamsters are solitary. A male shows up, mates with a female, and then he's gone and you barely even remember his name. What goes on then? The female, in the interests of leaving as many copies of her genes as possible, is hoping to have lots and lots of litters over the course of her lifespan. The male is only going to father one of those litters because he's long gone and somebody else is going to show up. What are the different incentives? The male would be delighted if he could make the offspring grow at such rates that they're going to be so healthy and they're guaranteed to survive and pass on copies of his genes. And if in the process this just metabolically damages the female so that her future reproduction is impaired, we got a conflict here. The male, the hell with your future, whatever, because you'll be doing it with other guys. I want to have my offspring grow as fast as possible. Female incentive is, let's slow it down a little bit to have a long-term strategy. Okay, so this sounds like economics, but what you see is there's a whole bunch of these bizarre genes called imprinted genes, which is a whole separate story, but there's imprinted genes from the father that makes the fetus grow faster. and there's imprinted genes from the mother that tries to slow down fetal growth. There's a battle between the two going on. And this is this whole separate field that's fascinating. So you look at that and say, oh my God, all of this conflict between the female and the male because they have differing goals and because they're not related. Oh, and suddenly you see things going in the other direction. Maybe, in fact, it's advantageous to mate with relatives because out of kin selection pressures, the intersexual competition gets lessened because you all share incentives to a much greater effect. So that produces a really bizarre suggestion that you instead mate with close relatives. Hey, so inbreeding avoidance, mate with distant strangers, getting the advantages of kin selection, mate with relatives, opposite directions. And some years ago, people did all sorts of theoretical models and came up with this prediction that the optimum for balancing these two directly opposite sort of forces on the system are third and fourth cousin matings. Ugh. So that's what the theory stuff showed. And what people have been seeing since then is it kind of works that way. First, in the non-alarming sector, there's all sorts of bird species where you see in mate choice, their ideal mate is a third or fourth cousin. And it's not just a function of they live near you because you're all related, because you got the first and second cousins closer. You can have even birds that disperse, and you see mate choice optimizes birds of lots of species. Their first choice is to mate with the third or fourth cousin. And when they do so, the likelihood of offspring surviving peaks for third and fourth cousin matings and goes down in each direction. Whoa, so that's birds. And then we look at humans who are not especially monogamous as a species, and that's a whole separate story. But we look at humans, and the same logic should show there. Avoid inbreeding depression and kids with 6 toes, and don't mate with relatives, and the kin selection advantages of shared goals with the individual you mate with because you share a fair number of genes. So is there any evidence for that? And there's one amazing slam dunk study now from 2008. It was published in Science, very prestigious, and it looked at a population on Earth, which is like God's gift to geneticists, populational geneticists, Iceland, the population of Iceland. Everybody knows what the original population was when it was first settled. It was an empty place at the time, and incredible Icelandically careful records for centuries now of who married who and who had kids and who survived. And what the researchers did was go back through 160 years worth of data and looking at fertility. The fertility that women had, how many surviving offspring did they have who survived into adulthood? And that's one of the sort of Darwinian measures of fitness. And what they observed was over the course of these decades and decades, over the entire population of Iceland, peak fertility is what you see coming out of third or fourth cousin marriages. So that's distressing and disturbing and all of that, but that makes some sense biologically. And that seems to be a pattern in the best studied inbred isolated population on Earth. Iceland, you see the exact same thing that you see in some other species, which is balancing inbreeding avoidance and advantages of kin selection. Where do those two opposing forces balance out? Somewhere around a third or fourth cousin. So that's odd and distressing, but that's what the science is showing.
And that's it for episode 56. Keep submitting your questions at the form found in the Instagram story highlight and bio or the YouTube video description. I'm Offspring Sapolsky, and thanks for your continued support of Science and the Beard.